Hi, I'm Todd Theriault, and this is the 12 a.m. hour of the 24-hour online event. And I'm joined today by Andreas Bueckle and Katy Börner, both of whom were instrumental in developing what we'll be talking about for the next hour, which is the VH MOOC course, the Visible Human Massively Open Online course. And they're going to take you through that, tell you a little bit about how it developed, and also show some videos from it. So with that, I'll pass it over to you, Katy.
Thank you, Todd. Thanks for having us. And yes, we are pre-recording. This is not midnight here right now. So for those of you which have not seen the Visible Human MOOC online site, anyone can access this course. You just have to go to this rather long link on the right-hand side. This is a 10-hour course that introduces the HubMap project to general audiences. And then over time, we added material from other projects, as you will see. And the ambition of HubMap is to map the human body from the entire male and female healthy adult bodies down to the single cell level and even down further to proteins and genes. And so it's really, really exciting to present this material, and we hope it will get more students enrolled in the course. And again, it's a free course, anyone can take it. The course itself tries to give a theoretical and practical understanding of different single cell tissue technologies, and you will see some exemplary videos that we will show you here. It will also help understand students how these single cell studies are actually done, how you do data harmonization across labs, across organs, and across technologies for suspension-based data, transcriptomics data, but also for spatially explicit data, proteomics data, and how you can build an atlas from all of that rich data that comes from so many different sources. The course does not only give you videos, it also gives you hand-on exercises where you can test your newly gained knowledge, where you can also submit your answers to questions, and then it's automatically graded. So you get hopefully a kick that you got it right. And then, of course, we also have been using this to onboard new team members. HubMap is a large project with 350 experts, all coming from different areas of research. So it's interesting to have them have a resource to get the same terminology, the same understanding of what is a map, what is an atlas. And that has been super helpful. At the same time, we also learned a lot making this atlas because we got to see and look behind the scenes. We got to go on lab tours. We got to see the challenges and opportunities that are faced not just by the computational teams, but also by the experimental teams. And ultimately, we get a better understanding of how a HubMap reference atlas or reference atlas of the human body might come into existence by joining forces with a lot of experts around the globe. So here, as you will see in a moment, we will give you an overview of the project goals, setup of ambitions, and in fact, we will run the official HubMap video. Andy is going to facilitate that. And then we will look at tissue acquisition and analysis, data harmonization. Ontologies are super important here, and we have a good video segment for you there. And then also challenges and opportunities for HubMap and other atlasing projects going forward. The instructors are myself, Alan Cordokus, and Andreas. And Andreas and I are here with you to guide you through this arm. So I will stop sharing, and I think Andy will go guide us over to the MOOC.
All right.
Thanks.
Hello, welcome to this hour. This is kind of a trip back down memory lane for us. We've been developing the VH MOOC for the past 5 years. When we made the first VH MOOC video, that was the end of the first year of HubMap, and I was in fact still a PhD student at the time, which has changed since then. I graduated since. So I want to now share my screen, and we're going to have a look together at the first video that we're going to cover today, which is the HubMap overview video. This video dates all the way back to 2019/2020. You will see a lot of live footage in there. We actually went to Stanford to the first or second HubMap all hands meeting. And this was my first opportunity at the time to meet all of the PIs, many of whom we still work with today. So there's a lot of like emotional value as well as scientific value in this video. This video is 4.5 minutes long, and I'm just gonna play this. Let me make sure I'm sharing my sound. And then I hope that we can all enjoy this bit of HubMap history together. Here we go.
No matter what age, race, or gender, every human body is formed of tens of trillions of cells, organized in a fascinating system which we only partially understand. Funded by the NIH, the Human Biomolecular Atlas Project, or HubMap, is a multidisciplinary research effort by over 200 experts in more than 50 institutions. Its goal? To create a map of the human body at single-cell resolution, a feat never accomplished before.
HubMap's a really exciting project. The challenges are dealing with data from a large number of tissue centers, many data formats, many assay types, and bringing those together, and then giving people tools in a way that non-specialists can actually do medicine and biology against this really unique, valuable data set.
One of the major challenges is the integration. I think there's a whole slew of disparate data sets and data types that are coming in. That all needs to be integrated. It needs to be registered and mapped, so to speak, to a organ system framework, whether that's a common coordinate framework or a physiological or biological-based framework.
To develop a map of the human body, NIH needs to coordinate efforts for a diverse international team. Challenges include heterogeneous data, remote communication, large-scale data transfer, and the standardization of workflows. All of this has to happen across a dynamic technological, social, and political landscape.
To put together a whole piece of product that we can deliver to the community and make them understandable to the general public while everything is somewhere in the middle of changing is very difficult.
At this point, we have 18 funded groups that we have to try to pull everybody together, get everyone coordinated and on the same path towards similar goals. Each group has a superpower.
Capturing cells in spatial, three-dimensional detail will help us bring together knowledge distributed across multiple disciplines.
If you go to a doctor, they look at you with clinical systems at the body level. If you talk to scientists, they look at the cellular level and they can explain you very complicated phenomena, but not necessarily are able to bring it all the way up to the medical level information. So what drove me was the idea that we need to bring this somehow together in a multi-scale, multi-dimensional database or tool.
My personal hope is that the work that we do in HubMap can then catalyze this as a general approach in many different areas of science. My ambition is that the legacy of HubMap in 10 or 20 years' time is really thinking about and realizing that using a single-cell analysis type approach for tissues, for understanding complex environments is the default way in which science is done.
So we're building a map on healthy human tissue, but ultimately this is going to be, we think, a model for what could be used for a human disease. What does it look like when things go wrong? So to have a nice, if you will, reference map that is incredibly high detailed on a healthy person, it's really going to help us understand what happens when things go wrong, when disease appears.
The need for a common language between several highly specialized disciplines prompts HubMap to become a catalyst for innovation and collaboration. Our need for knowledge has prompted many notable scientific advancements in the past. So our HubMap members are part of a great tradition, and they are only getting started.
This is really memory lane, Andy. I like it.
Yeah.
So if we go to the VH MOOC and we go and give everybody an overview of all the many different resources that people can access there, I think this would work well. So Canvas is used here as a platform to basically guide students through learning materials. This is a platform that's used for all of the courses at Indiana University, and it can also be used for these massively open online courses that allow thousands of students to come in. It's not always meant for thousands of students, but it can be supporting so many students. So here you have the first section you just saw. This is the HubMap overview, but you also see that it comes with a lot of references and image credits and data credits. So it's in the spirit of open data, open code, open research, very transparent with a lot of provenance data right there, and hopefully also provides a rich way to link into other existing resources, including also the software used to map science at scale here, as you just saw these networks overlaid over a map of the US. So if you go back and to the overview of all of the videos, let's just walk through basically October 2020. We published the very first modules, learning modules, the HubMap overview, which we just saw, tissue data acquisition analysis we go next to, and then further down you see more information on data harmonization, ontologies, portal design and usage, but then also open consenting your data, a plea from Mike Snyder to make data available for research, which I think is very, very important, because without contributions of healthy human tissue data, this project couldn't exist. And so this is core to what HubMap and other atlasing projects aim to accomplish. And then you see that there was a next second release of learning modules in June 2021, which also features an ontologies video, which we will show you in a moment, but also features more information on so-called ASCT plus B tables, which are core to capturing knowledge from experts. And they need to agree how to name anatomical structures, how to name cell types and biomarkers. Non-trivial, especially if it comes to many, many different organs with many different cultures and communities, which rarely talk with each other like they do here in HubMap and in other cross-organ efforts. And then you have more information on codex, which is one of the spatially explicit assay types to get single cell data. You get to learn more about Azimuth for transcriptomics data. And you also get an update, because as time goes on, the portal evolves, the data portal. And so more data becomes available, different types of data become available, new tools and user interfaces become available. Vitesse as a visualization interface to the atlasing data and to HubMap data, it becomes more and more advanced. Then the third HubMap portal release in June 2022, you have a halfway point, and I think we will show you a few segments from that. But you also have introductions to the Human Reference Atlas Common Coordinate Framework, and latitude-longitude system for the human body. You have using the EUI and the GTEx portal, which is a team we collaborated with at the Broad Institute. And it's now interesting to see that other efforts, consortia, are starting to use HubMap data, but also the Common Coordinate Framework and the atlasing framework with the ASCT plus B tables to make their data available in a new way. And then SENNet gets funded, which is the study of senescent cells. And in this project, we then also use some of the tools and the Common Coordinate Framework for harmonizing SENNet data. And so this is now bringing over some of the tools developed in HubMap to the SENNet community and making them very quickly and efficiently available to researchers there. Also adding registration of tissue to the SENNet data ingest process. And then going down, we need to onboard all the TMCs, which are tissue mapping centers. And so they need introductions in what in the world is a CCF, what is an ASCT plus B table. And so we added a lot of that information here. And then the fifth HubMap portal release in June 2023, more information on how to construct validated antibody panels, so-called OMAPs. And this is a video by Alan Quadocus, which couldn't be here today, but you might like to watch it later if you're working with immunohistochemistry assay types. On the next, further down, you see, maybe that's it. No, there's more. So there's a HubMap portal release yet again, because again, that portal starts growing, has more and more information. And maybe that's it, Andy?
Yeah,
That's
and then
all we have so far, right?
yeah, another thing to note is that as part of all of these releases, we also released updated video tutorials which are not listed in the VH MOOC, but which came out of these efforts nonetheless. They're just not listed here because they need to be updated frequently. But there was more video creation. This is just the stuff that's a little more evergreen.
And it's actually interesting because the data that evolves all the time has to go with the code, which evolves and gets better all the time, has to go with the training materials, which are featured here, which also evolve and get better all the time. So it's really, from time to time, it all has to sync, and then it gets published here.
Yeah.
All right, I'm ready for Spraggins.
All right, we're moving on to the next video, which is going to be Jeff Spraggins presenting the work of TMC Vanderbilt University. So I'm gonna
and that's a really interesting team. So we got to visit a number of times, got to see their vet lab environment. And their team has been phenomenal in creating highest quality data for different organs, initially kidney, but then also for other organs. And they also have an impressive way to then get three-dimensional molecular atlases of human tissue for these different organs and to make them freely, widely available to many. And so we, for instance, also use that data in some of the Kaggle competitions. And for me, it took a little while to load in the video, but that's neat to have here. And so if you go down, you will also get to see that on that page, you don't just have the video, you also have a lot of references to work by that team. And again, they're a very prolific team, which have been working on atlasing long time before HubMap came into existence, but also within HubMap have been extremely productive and collaborative. And then further down, I think you also have more data credits and image credits, software credits, websites to their environment. And again, I highly encourage you to check out these resources. All right, Andy, let's run this video.
All right, here we go.
Our tissue mapping center at Vanderbilt University is called the Biomolecular Multimodal Imaging Center. And it's a collaboration that includes Vanderbilt University, Vanderbilt University Medical Center, and Delft University of Technology. And the mission of our center is to develop a pipeline of integrated technologies that brings together imaging mass spectrometry with various forms of microscopy and traditional multiomics, with the goal of constructing comprehensive three-dimensional molecular atlases of human tissues. The focus of our center currently is on the human kidney. And we get those tissues either as discarded donor tissues or as tissue blocks from surgical resections. Once we have the tissue blocks, they're assessed by expert pathologists to determine their normalcy. And once they've passed those quality control checks, they can then go on to the full multimodal three-dimensional characterization pipeline. Now, one of the important things and unique things about HubMap is the focus on developing three-dimensional molecular atlases at cellular resolution. Now, for our technologies, to generate three-dimensional images, we have to take serial sections through an entire tissue block. So what we end up with are taking sections, sometimes hundreds of sections, and each of those sections is mounted onto a glass slide. So each of those samples are then run using different aspects of our characterization pipeline, some being analyzed by imaging mass spectrometry, others being analyzed by different forms of microscopy, and then we combine all of that at the end. Specifically, we're generating molecular images of human tissues using matrix-assisted laser desorption ionization, or MALDI, imaging mass spectrometry, autofluorescence microscopy, classical stain microscopy, and multiplexed immunofluorescence microscopy. We collect the autofluorescence on every tissue section prior to analysis with the other modalities. And the reason we do that is so that the autofluorescence can act as an anchor from one tissue section to another. So we have a common modality that spans all tissue sections. What this allows us to do is spatially co-register everything using the autofluorescence and ultimately allows us to link one modality to another in three-dimensional space. For the samples that will be analyzed by imaging mass spectrometry, first we collect autofluorescence microscopy. We then define specific regions on the tissue to collect various forms of MALDI imaging mass spectrometry data. And once that data has been collected, the nice thing about MALDI imaging is that the tissue remains intact. So following that analysis, we can then perform stains and collect classical histological images from the same tissue sections. For the samples that will be analyzed by multiplexed immunofluorescence, again, first we collect autofluorescence microscopy, and then following the autofluorescence, we then can perform the antibody-based staining and do that in a cyclical fashion to build up the number of markers for specific cell types within the tissue. Once all of the imaging data has been collected, As I said, we use the autofluorescence data to first register each of the modalities within a single tissue section. So for instance, registering the autofluorescence to the imaging mass spectrometry to the PAS stain microscopy, and create a co-register data set for an individual tissue section. We can also use the autofluorescence to construct our three-dimensional blocks and then project each of our individual imaging modalities onto that three-dimensional scaffold, allowing us to ultimately produce high spatial resolution, molecular three-dimensional images of human tissues, in this case, of human kidney tissue. As is the case with all HUBMAP projects, this is a very multidisciplinary effort that requires the creation and integration of many unique capabilities, including procurement and management of human specimens, development of analytical imaging pipelines, and ultimately the construction of bioinformatics workflows that allow us to visualize the three-dimensional molecular atlases that we construct and also analyze these large-scale multi-omics data sets. We believe that the data generated by HUBMAP will provide a new paradigm of understanding for the normal state of organs that'll span vast molecular and spatial scales, ultimately allowing us to make new connections between molecular profiles and cell types within human tissues.
Great. Yeah, some of this was done during COVID. So Jeff Spraggins actually did this out of his house. And we had many, many meetings where all of us were at home and working out of our home offices.
Yeah, so this is what when we produced lots of THM materials, we were like, there are two types of videos. There's type A and type B videos. Type A videos are the first one that you saw. Highly produced with live action footage, you know, a very classic kind of TV journalist kind of setup where you have a camera and an interviewer and lights and a PI sitting there being asked questions. That's a type A video. With COVID, that was no longer doable. So then the type B video is the kind of video that we're all very familiar with now, where it's somebody speaking over Zoom, giving a presentation. This one was a bit of a hybrid because we did some editing. You might have seen on the slides, you know, there are some highlights, and then we zoom into a specific part of this gigantic biomic workflow slide. So this one is kind of in between. But yeah, these kinds of videos were basically the best thing that we could do while we couldn't interview people in person. But yeah, this is definitely one of these evergreen videos.
And now we come over to ontologies, I think. And that's important because different experts might use very different names for the very same cell type. Some call it T cell, the other one just T because they're efficient. And so it's not easy to then bring order to all of this. So ontologies can really, really help here. And they also help connect the rich data from HUBMAP to existing knowledge frameworks and structures, and then ultimately you can reason over ontologies and connect it further out to diseases and to food and to drugs and everything else. And so it's really, really important to get that part right. And in fact, HUBMAP has been adding many, many new terms to existing ontologies so that they better cover healthy human data. And so here we get in ontologies one-on-one, and I think Andy has a few few items about that also, because that has an interesting history.
Yeah, so this video is a very interesting case because it is a kind of new type of video for us. It was produced almost entirely out of the house. Mark Mewson and his son, Jay Mewson, produced basically this entire video. There was very little work on our side, if I remember right. And it also shows that Mark Mewson is an excellent teacher, introducing the really abstract concept of what an ontology is in really straightforward and understandable terms. This is one of my favorite, if maybe not, it might actually be my favorite VHMOOC video in total, just because of the incredible informative value that it has and the ongoing value it has for all that we do in the HRA, where ontologies are a core part of what we do, where we use up to, I think, about a dozen ontologies that come into our knowledge graph. So briefly, I want to the site here. So on this side, we have the video, which we will look at in a second. And then this is a related video. Mark Mewson came to visit IU as an invited data science speaker in October of 2022, where he introduced his work at Cedar and at Stanford University for, you know, cleaning up metadata messes in experimental data. As per usual, we have a lot of references here and introductions. data credits and then software credits, and also additional websites for anyone who's interested in learning more about ontologies, which again are part and parcel of what the HRA is about. All right, with that, we're going to go into Mark Mewson's 13-minute video.
We all have memories of seeing our first real library, of encountering endless shelves of books and wondering how anyone could find what they might be looking for in such a large collection. How would you know if the library has anything in your area of interest anyway? Well, every book has a code. It might be a Library of Congress code, based on a classification system created in 1897. It might be a code from the Dewey Decimal System, created even earlier in the 19th century. We classify books on the basis of their content, and we use these classifications to find new information, to put books back in the correct place, and to assess a library's holdings. Classifications pervade our everyday life. Just like the Library of Congress, Amazon classifies its books to help us find publications we might be interested in, and, more important to Amazon, to bring our attention to books it thinks we might be persuaded to buy. All e-commerce sites categorize whatever it is they want us to buy. eBay has an online product catalog that is different from Amazon's. Craigslist organizes its content using an entirely different system. Each site on the web recognizes that its survival depends on how it categorizes its contents, and how well the terms that it uses match the way consumers think about the products that are being offered. It's not much different in biomedicine. Biologists may not be trying to sell anything, but they often need to categorize the things they study and to group things together to clarify their common properties. We call this categorization an ontology. Ontologies are often visualized using an indented tree structure, as in a file browser. This ontology of amino acids, developed at the University of Manchester, tells us that alanine, along with asparagine, cysteine, and glycine, are some of the aliphatic amino acids. A biologist can examine this ontology to know what amino acids exist, what their properties are, and how one might classify them. An ontology may exist only as a conceptual scheme in our heads. We all categorize and classify the things in our world, but we seldom are explicit about writing down our classification systems. When we do write down our ontologies, they become available both to people and to computers. If we want to know what kind of amino acid alanine is, or if we want to write a book about alanine, or if we want to state that we performed an experiment to measure the concentration of alanine, then ontology can give us a set of standardized terms and relationships that can facilitate all of these things. Our ontology can tell us that aromatic amino acids are not aliphatic, and that amino acids have other properties, such as a standard genetic code. Biomedicine is simply replete with ontologies. Nearly 300 years ago, Carl Linnaeus proposed his binomial nomenclature for speciation. Linnaeus's hierarchical structure of categories of life forms, with each class of organism having a unique genus and species, was a seminal accomplishment. It's still with us today, and perhaps the best-known scientific classification. In the 19th century, the precursor to what is now the International Classification of Diseases, or ICD, appeared on the scene. The ICD provides an ontology of diseases that the World Health Organization uses for international public health activities, and that the United States uses for all medical billing. By the turn of the 21st century, a huge international effort was underway to develop an ontology to represent the properties of gene products, the biological processes to which they contribute, the specific molecular function that they perform, and the cellular component with which they are associated. The result was the gene ontology, perhaps the most widely used ontology in bioinformatics. The gene ontology is instrumental for interpreting many high-throughput experiments, and it provides a comprehensive, well-curated digital knowledge base of basic biology. So ontologies are everywhere in biomedicine, standardizing the way that we refer to basic biological processes, to human diseases, and to everything in between. The whole idea of ontology is credited to Aristotle, who formalized the notion of categorizing what exists in the world. Ontology became the branch of metaphysics concerned with the study of existence. In recent years, however, the study of ontology has leapt from the realm of philosophers to that of computer scientists, who create data structures to encode what entities exist in some application area, what the properties of those entities are, and how the entities relate to one another. The desire of biologists to use computers to catalog the results of gene expression is not far from that of the software engineers at Amazon who refine the company's product listings. We are all engaged in work to enumerate what exists in our respective domains, to clarify and communicate properties and relations. We do this to create a description of what exists in some particular application area for the benefit of people who seek clarity regarding the structure of some human experience, for software agents that may need to process information about some application areas, and perhaps most important, for the interaction between people and computers, so that people can be explicit about what they mean, and so that computers can act on human input in the most precise manner possible. We create ontologies as standards, so that we can have agreed upon terms for describing the things that matter to us. There is no need to redefine speciation, for example, when Linnaeus already solved that problem. Having such standards allows interoperation. I can compare my data to your data if we call things by the same name. Computers can also reason about things reliably and efficiently when we use standardized terms and IDs to name things in the world. There's another side effect that occurs when we develop ontologies to describe the entities in an application area that matters to us. We solidify scientific communities when we all use the same language to refer to the same phenomena. Science advances when we can agree on what exactly we are studying and what we should call things. The HubMap project is a large undertaking supported by the United States National Institutes of Health that relies heavily on biomedical ontologies. The goal of HubMap is to create an atlas that can describe every type of cell in the human body, and to describe the characteristics of cell types in terms of genomic, proteomic, and other biomarkers. HubMap needs to be precise in how its atlas talks about the anatomical location of cells. It needs to be precise in how its atlas talks about cell types and biomarkers. There is no other way to obtain the necessary precision than to use standardized ontologies. When scientists who are working on HubMap think about collecting data about an organ such as the kidney, they recognize that the anatomy of the kidney includes a set of substructures called calyces that are all wrapped in an outside layer called the renal cortex. Each of the calyces has constituent parts that are understood all the way down to the cellular level. The HubMap investigators are capturing information about the kidney and about many other organs in the body in a format that lays out the anatomical substructures, the constituent cell types, and for each cell type, the known biomarkers that identify cells of that type in the particular anatomical location. These tables for anatomic structures, cell types, and biomarkers are known as the ASCT+B tables, and they depend heavily on ontologies. The anatomical structures need to be represented explicitly, as do the cell types and the biomarkers. The use of standard ontology terms for all the components of the ASCT+B tables enables scientists to describe anatomical structures, cell types, and biomarkers with precision. People can search the tables using the ontology terms. The use of ontology terms allows them to be confident that they can access all of the relevant data and that they can evaluate all of the information that has been stored. The ASCT+B tables represent anatomical structures using terms from an ontology for vertebrate anatomy known as the Uber Anatomy Ontology, or Uberon. How do you get to see what terms the Uberon ontology provides? There are several repositories that store information about biomedical ontologies. The most comprehensive is a system known as BioPortal, which has been supported by the NIH to provide access to all publicly available biomedical ontologies in the world, currently several hundred. We can use BioPortal to search for the Uberon ontology and see that Uberon consists of more than 20,000 classes or terms, and that users view Uberon at the BioPortal site nearly 1,000 times every month. BioPortal can show us all the terms in Uberon in a tree browser. If we want to focus on what Uberon has to say about the kidney, we can search for that term, and BioPortal will bring us there. We can see that Uberon has a term for the kidney, and subclasses indicating the left kidney and the right kidney, as well as terms for the progenitors of the adult mammalian kidney that are seen at different stages of embryogenesis. Uberon makes distinctions about anatomy that are different from those made by another ontology of anatomy, the foundational model of anatomy, or the FMA. We can visit the FMA in BioPortal and see that it includes more than 104,000 terms. The FMA is also frequently viewed by BioPortal users. The FMA includes terms for the kidney, as well as the left kidney and the right kidney. The FMA models only adult morphology, so it doesn't list the terms for the embryonic kidney structures. In fact, when we compare the FMA with Uberon, we see some interesting differences. Uberon lists the embryonic structures, whereas the FMA does not. Uberon models the kidney as a kind of cavitated compound organ, whereas the FMA models the kidney as a kind of parenchymatous organ, which is a kind of solid organ. Which ontology is correct? Well, that depends on the assumptions that the ontology's developers have made, and whether those assumptions apply to a user's situation. We can't always tell by inspection what those assumptions may have been, and thus why ontologies can make different or even contradictory statements about the same entity. One ontology might model light as a wave, and another might model light as a particle. Every ontology developer has to make assumptions about the context in which an ontology will be used, and those assumptions often govern what an ontology will say about the world being modeled. Ontologies are thus models that, within a particular context, enable us to talk about the entities and relationships within an application area without ambiguity. But ontologies do not necessarily share the same perspective. One ontology may talk about the adrenal gland, and another may talk about the suprarenal gland, but they are really talking about the same thing. However, sometimes the kidney is represented as cavitated and sometimes as solid, and we have to accept that the context in which an ontology will be applied can affect how we characterize the entities that may be modeled. The major contribution offered by ontologies is that they standardize the way we talk about the world. The gene ontology provides standard terms for describing gene products and their behavior. Uberon and the FMA both standardize terms in anatomy, but they do so in slightly different ways. Both ontologies continue to evolve as we learn more about anatomy, and as the developers make new decisions regarding how to model anatomical structures. Biomedicine has hundreds of ontologies that describe different areas of interest. The availability of so many ontology resources offers a great benefit to biologists and clinicians, although the total number of ontologies can sometimes be overwhelming. Fortunately, resources such as BioPortal can help users to identify the ontologies that are most suitable for a particular task, and applications such as HubMap's ASCT plus B tables can draw on these ontologies to standardize their terms, to ease access to complex information, and to help us all to speak the same language when we interact with these computer systems.
Yeah, he is a great teacher, and his son is a great presenter. Really pleasant
Great duo.
to listen to that. And yes, the Human Reference Atlas that is developed within a HubMap in close collaboration with 20 plus other consortia now is published via the BioPortal, so you can access it there. And it's also now published via EBI, which is institute in the UK. It's a Shanghai Institute, and we are working on different mirrors that will have that data around the globe so that people can access it and add their own local data to it and then share back globally. And BioPortal now has, I had just looked, it has 1,166 ontologies. So there are many ontologies out there, and a total number of 15 million classes and 36,000 properties. So it's really a massive knowledge graph of what we know about human biology and other biology. There are lots of other species in there as well.
Another fun thing that gets me every time I watch this video is if you look at Uberon, it's a multi-species ontology, so it doesn't just capture humans. So a couple of like spots above the kidney is the ampulla of Lorenzini, which is in sharks and stingrays. And so I just find that funny how, you know, in an ontology, because it's this tree structure, you sometimes have completely different animals very close to each other just because, you know, of the structure of the organs. There's always a little thing that gets me about this video.
All right, so let's look at what the CCF actually is. And I think we have the introduction to the HRA and CCF video from 2022, still good.
What makes HubMap's registration user interface and exploration user interface able to handle a rich diversity of data from a broad variety of data collection methods? To answer that question, we need to understand how both programs leverage a common coordinate framework created specifically for the Human Reference Atlas, the CCF HRA. The CCF HRA is a computational framework that combines expert-curated ontologies for specimen, biological structure, and spatial data to create an underlying reference map of organs, tissues, and cells in the human body. The RUI and EUI use the CCF HRA to describe collected tissue blocks and biopsies in a standardized way. This enables data harmonization across consortia, donors, organs, and assay types. As of May 2022, the CCF HRA's 3 ontologies have been fully implemented for several organs, including the kidney, which is shown here. This enables the RUI and EUI to capture who, what, and where information for every tissue data sample. The WHO information, specimen donor metadata, is integrated and organized with the CCF HRA specimen ontology. To create this specimen ontology, the more than 100 metadata fields covered by HubMap clinical data was reduced to a smaller set of fields most relevant for CCF HRA construction and usage. The specimen data was then compared with data made available by other consortia to finalize the field selection. Currently, CCF HRA specimen data includes demographics and clinical data, workflow information, author information, and links to source data. The what information is captured by the CCF HRA biological structure ontology. While organs differ substantially in their form and function and across human specimens, all have anatomical structures, cell types, and biomarkers that are captured in the ASCT+B tables and the associated 3D reference object library. The resulting ASCT+B tables represent expert knowledge and information from textbooks, scholarly publications, and experimental data evidence. An associated 3D reference object library captures the anatomically correct 3D shapes, sizes, locations, and rotations of key AS present in the ASCT+B tables. These reference organs are developed by specialist 3D medical illustrators, and they are approved by organ experts. Finally, the CCF HRA spatial ontology provides the framework for the where information. Spatial data describes the 2D and 3D shapes of entities and their physical size, shape, location, and rotation from the whole body level down to single cells. A unique feature of the CCF HRA is the interlinkage of the ASCT+B to the 3D reference organs, which supports new forms of semantic and spatial queries. For example, given a registered tissue block, the 3D anatomical structures it contains or collides with can be determined and used as annotations. Using these AS annotations, all AS and CT inside of these can be retrieved. It is also possible to use cell types as search queries and to retrieve all AS that typically contain these CT.
Yeah, and this has evolved since then. I just checked, and we now have 5,577 tissue datasets mapped into the Human Reference Atlas from 439 donors. So this is really a massive undertaking across many different consortia, many teams contributing to this atlas.
We also have almost 1,100 tissue blocks now. So that's quite a massive upscaling since. Also,
Yeah.
Todd, that was a great job you did with that video.
Thank you. I was very concerned to get it right, and it was a lot of information to pack into a 4-minute video. And it's one of those things where the more that I worked on it, the more that I understood the immense challenge that had to be met for all of these working parts to talk to each other and to be collected, and the data and different types and all of this. I thought, how does this high wire act ever get pulled off? And that's one of the nice things about creating videos like this, is you really have to kind of overstand the material a little bit to recreate it and interpret it for a general audience. And so that was really very rewarding to do that.
The other thing I think that works really well about this video is it brings together the ontologies, which we talked about in Mark Newson's video, with the actual 3D interfaces.
Mhm.
These are two completely separate worlds, but the HRA
Exactly.
brings those together by saying, hey, under the hood, under all of these nice 3D reference organs, like the liver in there with the nice lighting and the Google viewer, underneath all of that is actually ontologies, and each of these anatomical structures is annotated with one of those. I think that's what this video demonstrates in a really, really strong way.
Absolutely, and I think it's good to see the difference between these kind of prosaic looking Excel tables, right? And they're so important, and then see how then this is also it's absolutely dependent to have these 3D organs, which are so beautiful and so dynamic, but they're dependent on this kind of just straightforward Excel table data. So that's amazing.
Yeah, I mean, as Jeff Spraggins mentioned earlier in his talk, one of the special things about HapMap is that we're building these molecular maps in 3 dimensions.
Mm-hmm.
And so that's when, having these 3-dimensional reference organs is even more important.
Yeah.
And there's an entire panel on why 3D as part of this 24-hour event.
Right. Yeah.
And
Tune in at 8 a.m. Eastern time.
Mm-hmm. Yeah. Please come back.
All right. With that, it might be time to go on to the next video. The next video is going to be the HapMap halfway point. So this is a module that we deployed in the summer of 2022. This was the end of the first 4 years. So HapMap is 8 years. This was the end of the first 4 years. We hosted a HapMap demo day here in Bloomington, Indiana at IU. And as part of that, we had many of these PIs that 3 years before we had interviewed at Stanford, we now interviewed again. And some of these PIs were new. And so it was just a really, I would say, heartfelt video production. Again, it was again a type A video, where we had people come in and we set up a nice little TV studio, and Todd did a great job interviewing. I was behind the camera and Katy was sending us people. So it was a really good production experience, I would say. And we're going to get to that later. So yeah, the March 2022 HapMap Demo Day here at IU Bloomington. Enjoy.
It's really awesome to be able to look back and say, we've started with this group of people who have never worked together before 4 years ago. And we've now formed working groups, we've formed functional groups that are working on data portals.
Probably the major accomplishment of HubMap is just bringing this consortia worth of people together. I'm a biologist, but I am not a computational scientist or a data analyst, or I know what I want something to look like maybe on a website, but I certainly can't do that.
Yeah, so my main achievement I think is the validation system for the ASCT plus B tables, and I think that that system prompted me to think about how we could make more accessible views of the ontology products we build.
I think when I last looked, and this might have been last week, we had over 50 papers on bioRxiv for HubMap. And it's pretty awesome to be able to watch some of these folks just churn these papers out.
One of the big challenges is just collecting the data that we really need to now integrate all those data together to build the reference maps that we're looking to build that connect the molecular information with the spatial information across all these different types of molecules and assays. And that's a socio-technical challenge of setting expectations, of establishing standards in the consortium, and then doing it in a way that is win-win for everyone involved.
You're integrating data from many small labs. Other projects I've worked on, the standards have kind of emerged because of the dominance of very big institutions. And I think there's that balance between, like, I've got a minimal viable product and this is what I'm going to do in the next cycle of 4 months, versus I'm building a sustainable structure that is extensible and is going to be able to do all this integration work to build these cool tools. But every time that has to be focused on really clearly stated use cases, otherwise I think you're sunk.
There were certainly a lot of things that turned out as we expected in terms of data types that we could generate well, we could get lots of data, but could we also get groups who weren't necessarily the developers of the technology to also generate these data types? And I think we were pretty successful with that. We also ended up developing a number of new technologies along the way, which I think will be important for the next phase of HubMap. The next 4 years of the program will be what we described as being the production phase. So we'll prioritize generating data types that we know we can generate well, that we know how to integrate them into some model of the human body. And then the fourth phase is really in the last year of the program, which is how we think about transitioning to some kind of sustainability.
I do want to see it more accessible, both for education and just for the person that's coming in because they're interested with this particular cell and they want to know how it changed from organ to organ or age to age.
As we're here at this demo day at the end of the first phase of the project, I see we're making progress towards that goal of starting to align with the work that Katy and Rahul are doing to establish the framework for building these reference maps and connecting the data together in a way that will enable us to do that.
And something that really shows a proper linked open data approach where you can go very rapidly from I want to query the semantics, I want to query the metadata, to this is giving me a piece of data that I can do useful work with. I think that's the dream. Right now I'm going to sort of find data sets and then now I've got to mess around with how do I actually get to the data? Do I write to the person? Do I follow a link which then tells me to follow another link which then might end up at a dead end? Do I have a license? All of that stuff, that can be made frictionless.
So my dream in this time is to be able to establish a sustainable framework by which not only future projects that may follow HubMap, but also other projects throughout the world and other consortia that are working on similar problems can follow to one day have that full complete map of the human body at cellular resolution. That's not going to happen in 4 years.
I get the cool opportunity to be able to brag about all of the really awesome work that the scientists and the researchers and the technologists are doing. They're building pipelines and they're putting all of these really awesome tools together, and I get to tell everybody about it, my team, and that's really fun.
Very fun, very fun. Yeah, it was good checking in with all of these folks. Many of them you saw in the first video. Others, they only joined halfway through the first part of HubMap. So it was kind of their first interview in the course of this VH MOOC as well.
But I think it's also true now that you can go to the HubMap data portal and get a lot of data, especially spatially explicit data. So HubMap is really unique in that. And then you can go over to the HIA portal and you get to explore the atlas and all the many data sets that now have been mapped into it. And so at the very end, we might show the dashboard for the HIA portal. And that gets you a lot of information on how that is used now around the globe. But I think Andy had a very special video that we also wanted to make sure you saw, because it shows that it's all work and play. I mean, it is a lot of hard work, but it's also fun. It's a great team.
Yeah, we had many hours of video interviews on top of many hours of Zoom recordings. And I organized all of my editing in my editing software. And whenever sometimes you just have funny little bits, like 4 to 5 second bits that you're like, this was really funny, completely unintended. Maybe somebody pronounced something wrong, in good nature. Maybe somebody, kind of was nervous, and then you get a blooper. You might be familiar with the concept of bloopers from TV shows or from movies. So we made a VH-MOOC blooper reel. It's 1 minute and 29 seconds long, and we edited that at the very end of the VH-MOOC, and I want you to enjoy it here with me. So here we go.
This number of glomeruli.
I'm recording a video for work.
Referred to as calling curves. Nope.
And Katy will have a better answer, so.
Add to the statement.
Can I ask the final question?
You said the previous one was the final one.
Andreas introduced as always the next question.
All right.
Right, ready?
Yeah. HubMap will create. Can you do again?
Now you're in the frame. You're in the frame.
I was trying to think of something funny to say, but I'm very unfunny.
Creator uploaded.
Into that kind of exciting time where we can really brag about it.
Okay. So on this dark desert highway, cool wind in my hair.
I have one I realized we never got to the hot dog though. We have to back up at some point.
This one is a UMAP plot from the salmon RNA-seq thingamabob.
And that Richard would go with it.
This could either be an outtake or it goes into the movie. Let's wait. What always gets me is Mike Schneider's bit with the sausages. So the story there is, when we made these interviews, we contacted people beforehand because we had to schedule these interviews and get a space at Stanford. And we told them, hey, please bring an object of interest that you can show to the camera to kind of, foster some engagement and make people, be interesting in front of camera. Sometimes people get nervous and they're very much inside of their shell. And so if you ask them to bring an object, and Mike Schneider, he came there with a kielbasa and a smaller sausage to demonstrate how the small and large intestine connect, because that's what he is studying. And so that's just an image for the ages. All right.
Yeah, and there's actually an entire video on that, right, which you can still look at. So just to conclude this, there is a dashboard now, the Human Reference Atlas portal dashboard, which gets you information on the data that now exists, how the data is used, how publications are mapped onto the Human Reference Atlas as kind of scholarly evidence, how experimental data, almost 200 million cells have been now mapped onto the Human Reference Atlas, and also diversity inclusion, which we take seriously. We want this to be an atlas that's relevant for male and female, many different ethnicities and age groups. And it's important to get that right. So please check out the portal. And then, of course, please come over and join us in the Visible Human MOOC. We are adding new materials all the time, as you see, and we would love to see you there. And then you can take messages home, bring family and friends, and also other colleagues. And also, please do consider using it in your teaching. This is open, free training material. This links to a lot of open, free data and code, and we would like to see that used in classrooms and beyond.
Thank you, Andy and Katy, for this overview of the VH MOOC. Hopefully your appetite is whetted and you head there. We still have another 11 hours left and lots of good stuff still to come, so stay tuned. Thanks again.
Thank you, Todd.